Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled MA900 Cell Sorter, Increasing the Capability of Multi-User Labs by Combining Automation and Technologies to Support Multi-Application Needs. My name is Blake Foreman. I'll be moderating today's presentation. I'm delighted to be joined by our guest speaker, Anthony Carcio from Sony Biotechnology. In this webinar, we will be covering how an automated and streamlined sort setup can increase the capabilities within a multi-user lab, how the MA900 enables seamless setup and exchange of different chips without instrument downtime between users, and how the MA900 system can be maintained in multi-user labs for successful, high throughput, and gentle sorting. Following the presentation, we will have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit these to the left of your screen. Simply hover over the questions icon and click to type in your question. Without further delay, I'd like to hand over to our speaker. I'd like to thank him again for presenting for us today.
Thank you so much for that kind introduction. Today, I want to share with you the key aspects and benefits of the MA900 cell sorter that make it an ideal choice for a multi-user and a multi-application environment. So in this presentation, we'll talk about some key considerations for cell sorters in multi-user labs. We'll talk about the technology at work in the MA900 cell sorter. We'll talk about automated and streamlined setup for sorting with different types of chips. We'll talk a little bit about troubleshooting and how easy that is on this platform, how to resolve some familiar issues. And then we'll talk about the multi-application capability and the performance for sorting with different cell types. So let's talk a little bit about the evolution of cell sorters. Flow cytometers and cell sorters used to be these, behemoths that filled a room and required a very specialized operator with a lot of experience and expertise to run them. And you'd be able to detect, maybe one, maybe two colors. Over the years, cytometers and cell sorters have become significantly smaller, much more capable, and significantly easier to use. So what is a multi-user lab? What is a shared resource facility? Let's talk about that. So a multi-user lab, that is a lab space where an instrument may have been purchased for everyone in the lab to have access to and share the use of. There's not always one particular person in charge of the instrument, and oftentimes trying to establish and follow self-written SOPs or paper SOPs near the instrument can be challenging. And it's going to be used and maintained by different people all of the time. In a shared resource facility, it's similar, but there's these are often called shared resource labs or core labs, and these are labs that are maintained and staffed oftentimes by an institution for the purpose of multiple labs or multiple departments using and having access to the instrumentation and the expertise of the staff. Most shared resource labs or shared resource facilities are run in a fee-for-service type business model, and they're often overseen by a director or manager or some type of other administrator that's overseeing the day-to-day operations of the lab and of the instrumentation. However, there are several key considerations for both of these types of labs, right? They're both these multi-user type labs. They both support many users with varied expertise and applications, right? And training these new users can oftentimes be very difficult and time consuming. On top of that, some labs often require 24/7 access to the instruments so that their users can sort their cells basically when they are ready or when their cells are ready, day or night. And so in both of these types of instances, an effort is typically made to minimize or streamline the time between users in order to facilitate their science more effectively and allow for maximum instrument access. As well as minimizing maintenance and troubleshooting and just downtime in general, because especially in a shared resource facility type of lab, the goal is always to increase lab efficiency and productivity. Okay. So some diverse applications with complex cell types, anywhere from cell lines to nuclei, immune cells, yeast cells, other types of bacteria, neurons. This is not an exhaustive list, but these are all types of cells and applications that we've seen run on cell sorters. So basically, what does the workflow look like in a shared resource lab like this or in a multi-user lab? Typically, the cell sorter is started up by some kind of administrator or staff or the first user of the day. They run their experiment, right? Maybe in this case, they sort with one type of nozzle. Then they do some kind of clean. Another user comes in, wants to run a different application or a different cell type. And then at the end of the day, the last person who used the instrument is responsible for cleaning and shutting down the instrument and any other type of maintenance that might be required to keep that instrument going. So some things that instrumentation in this lab would be useful to have. Walkaway procedures for startup would be ideal. Automated setup and system calibration so that somebody could just start up the system, walk away, come back when it's ready, and guided troubleshooting as well. So if there's an issue during startup or during the run at all, what do you do? How do you resolve the issue? The ability to sort different applications on the same instrument, right? So you can have one instrument that can run multiple things, and you can adapt it to different cell sizes. And because it's running different applications and cell sizes and you're running it daily, you want that performance to be robust. You don't have to worry about the variability day to day. And then finally, when you're cleaning and maintaining that instrument, you want some kind of guided workflow for how that's done, right? Especially in a multi-user environment, if there's not one person that's responsible for this, they may not always know what the protocol is for maintaining that type of equipment. So the MA900 cell sorter was introduced about 3 years ago with this multi-user, multi-application environment in mind, right? So it has a very small footprint. Installation on standard laboratory benchtops is typical. Has an optical configuration of up to four lasers and 14 parameters, so two scatter, forward scatter, and back scatter, plus 12 fluorescence parameters. And it's capable of four-way tube sorting and plate sorting up to 384 well plates with index analysis built in as well. And finally, up to 3 sorting chip sizes. And we'll talk about the sort chips in a moment. Again, sort of ease of use is always at the forefront of everyone's minds in these types of environments. So, these optical configurations are user configurable. It's easy to switch between, tubes and plates, and it's easy to switch chip sizes. So we'll talk about all of this. Additionally, because of, you're in this multi-user environment, some of these applications may require biosafety considerations, right? So the MA900 can actually fit into a custom Class 2 biosafety cabinet that provides product, personnel, and environmental protection during all stages of the sorting. You have basically 2 forms of protection. You have an aerosol management system from the sort chamber, as well as the laminar flow of the biosafety cabinet itself, and that ensures maximum operator protection. Okay, so the heart of the MA900 is really this microfluidic sorting chip. This sort of forms the core of the fluidics system. So this chip is both your flow cell and your nozzle, and it's available in different sizes. It is aligned to the laser daily for optimal performance, so you don't need to worry about shifts in alignment over time. Every single time you put a chip in, that chip is going to be aligned to the laser automatically. And so over time, your alignment is very consistent. The chip can be used over a 24-hour period for multiple sorting samples and can actually be exchanged in between samples as needed by the application. And this allows you a great deal of control over sample carryover. If carryover is a very big deal for you, you can simply pop that old chip out and put a brand new clean chip in and not have to worry about carryover at all. It also allows us the ability to easily recover from air bubbles and clogs without actually having to take the chip out of the instrument. So if you've worked with cell sorters in the past, you know that air bubbles, clogs, this is always a concern when running these types of machines. And if you've ever dealt with clogs or air bubbles, they can be kind of a pain and oftentimes requires you to take out the nozzle and sonicate it and clean it and restart calibration and all of that. The chip does not need to be removed from the instrument and can actually be, you can perform troubleshooting right from the software at the click of a button. Okay, so what are we looking at in this chip? So we have our sample line input, which is this sort of bottom port here. One more port up is where our sheath fluid is injected. It will go flow into here and flow down both sides of these microfluidics channels here and meet in the center. At the very bottom is the nozzle. So this chip will actually be vibrated with a piezoelectric transducer, which will form our individual droplets. At the very top, we have this vacuum port. This is what actually enables us to do the removal of air bubbles and things like that from the chip without taking the chip out of the instrument. You can simply backflush the chip, backflush those air bubbles out, and then resume the flow, again, without removing it from the instrument. And then finally, the laser intersection is right here, right above the nozzle. So you have your sheath fluid flowing down, your sample being injected into the center of that sheath fluid, forming your core stream, and then your laser intersection happens here, right where your laminar flow is, and then your nozzle. Okay, so how does the microfluidics chip work within this fluidics system? Basically, like I said before, it's the core or the heart. So you have a sheath tank full of your sheath fluid. This sheath tank is pressurized, forcing sheath fluid through a 0.2 micron sheath filter into the chip. Then you have your sample that is loaded into the sample chamber. This entire chamber is pressurized, which forces your sample up through the sample line, through a little pinch valve into the chip here. And then as these fluids flow down out of the chip, you have your nozzle at the very bottom, like we discussed. Piezoelectric transducer vibrates the chip, forming your droplets, and then your droplets are charged with your targets of interest. And as they fall down through the deflection plates, they are deflected into your collection tubes or your plate, whatever it is that you're sorting into. And then in the center, you have a little waste collector that is catching your empty droplets and your droplets containing your non-targets or the cells you don't care about. Okay. So these microfluidics chips come in 3 sizes, like we mentioned. This enables a very large range of cell types to be run on the MA900. And these chips are all essentially the same. All the microfluidics are the same. The only difference is that at the very bottom, the nozzle size is different. So 70 micron, 100 micron, and 130 micron are the choices available. And if we look at some specifics of these, basically the 70 micron chip is the smallest, so you have the smallest droplets, but allows you to sort the fastest. So these are used for small cells, typically bacteria, yeast, lymphocytes, things like that. Then as you go up in size to the 100 micron, droplets are a little larger. These are used for cell types like macrophages, CHO cells, fibroblasts, other types of, medium to larger cells. And then as you move up to the 130 micron nozzle, this is what's used for the largest cells or the most fragile cells. The droplets will be significantly larger, but you're running at a smaller or a lower pressure, right? So if you have cells that are very fragile or don't like going through the sorter, this is a great nozzle choice or chip choice. So things like HeLa cells, HEC293T neurons, these are cell types that typically do well with the 130 micron nozzle. Okay, so if you've ever used a sorter in the past, you know that there are sort of all of these different elements of the sorter that need to be set up and calibrated to ensure a successful sort. So you need to make sure that you have a proper optical alignment, that your lasers are aligned to your flow cell properly, so that as your cells are flowing through your flow cell, you can detect them. If you have multiple beam spots, you need to calculate the laser delay so that when you look at those cells in your software, all of the measurements are appearing where they should. You need to make sure that your droplets are calibrated properly so that you have a properly established break off, and then everything is nice and stable, and those droplets are going to be able to effectively carry your cells to their final destination. You also need to calibrate those side streams, right? So as those droplets are charged and deflected, we need to make sure that they're actually being caught in the proper location. So whether it's tubes or plates, whatever it happens to be, we want to calibrate the angle of those side streams effectively so that they're not just spraying all over the inside of the machine, or worse, going into some place you don't want them to, and now you don't have the purity that you thought you did. And finally, we need to make sure that we're charging the droplets we think we're charging, right? So we need to make sure that once that cell is detected in the flow cell at the laser intersection point, we need to make sure that the instrument is charging that droplet at the breakoff so that when we identify our target of interest, we know that we're charging that droplet and then throwing it into our tube or our plate effectively. Because if this is not calibrated properly, then we're charging empty droplets or droplets that have non-targets in it, and we're not getting what we think we're getting, right? Okay. So the MA900 is going to streamline the setup by automatically calibrating all of these calibration tasks for you. The sorting chip is user exchangeable, easily installed, and will guide you through the process using software wizards. So from the moment you log into the software will hold your hand and tell you exactly what to do. You do not need to know anything about, pressure settings, droplet frequencies, any of those specifics. The chip will actually have a QR code on it that you will simply scan using the camera, and then it will tell the software based on the chip size what pressure to set up at, what droplet frequency range to set up at, and all of this will be taken care of by the software. So inserting the chip is very easy. It looks something like this. Simply place the chip in, and then the instrument will pull the chip inside and begin aligning it to the flow cell, or aligning it to the lasers, I'm sorry. So again, this is important because it enables the MA900 to be an instrument where a user of any experience level can walk up to the instrument and begin setting up for a sort without having to have a high level of experience. Okay, so like we just discussed, step one, start up the sorter, scan the QR code of the sorting chip. The QR code will automatically input the baseline settings, and then the actuators will ensure precise positioning of the chip in the chip loader. Step two will actually be loading a tube of auto calibration beads. This is what is going to enable the automation of all of these different tasks. And so that will look something like this. Got these beads, scan the chip. And what's important about these beads is they do not need to be diluted. So again, very easy to use. Just shake up the tube, add the tube of beads, and then once that tube of beads is loaded, the software will begin setting up all of these different steps, right? So chip alignment will be done automatically. It will move the chip in the X and Z direction to align it to the lasers. It will calibrate the droplets, making sure that the break-off point is where it should be and that those droplets are nice and stable. It will then look at a couple different angles of side streams and ensure that it chooses an angle where it will deposit those droplets into your tubes or your plates. And then finally, it will do a sort delay calibration in order to ensure that it is charging the correct droplet. And again, all of these steps are fully automated. Once the process has completed, it will tell you that it's been completed, and it will sort of give you an image of each of those steps. All of these steps are also recorded in the software logs. So if you ever wanted to go back and see what a successful calibration looked like or what a failed calibration looked like, you can go into the logs and see all of that. All right. So like we said, automation provides ease of use and a walk-away advantage, right? Because once you load that tube of beads, there's nothing more for you to do. I always tell my customers once those beads go on, go ahead and go get coffee, finish prepping your cells, take a meeting. Truly, there's nothing else for you to do until the instrument finishes the calibration. And this setup is precise, accurate, and consistent. Because you're loading a new chip every single day and that chip is being aligned to the lasers, you don't have to worry about the instrument coming out of alignment. You don't have to worry about what did the last user run through that flow cell and is it all, scratched up or dirty or clogged. We don't have to worry about carryover. And it's a streamlined operation for all the users, and it really eliminates the intensive training requirement because, again, this is just a wizard that from the moment you log in, it's going to tell you exactly what to do. And actually, that's all great, but what happens if something does go wrong, right? So let's say it's in the middle of setup and it gives you some kind of error code. It will always give you the error ID number, and then we'll actually give you a little troubleshooting, basically like a sub wizard, right? So based on what the error code is, if you click on the troubleshooting button, it will then actually give you another wizard where it says, okay, you should, do a sample line cleaning or check to ensure that you added enough beads to the tube, whatever that error code happens to be. So truly, walk away setup, and in the event of an issue, it will help you with that as well. So the important part about this is that even when there is an issue, you don't have to take time out of your day to sort of deal with that. The user can deal with it themselves. Okay. So that's all fantastic. My sort's ready to go, but how do I know it's going to remain stable? Do I have confidence that I'm going to be able to sort for a while on this instrument and know that everything is okay? In fact, yes. So the system will maintain and will monitor and maintain not only the calibration of the droplets, but the temperature as well. So if you ever sorted before, you know that those droplets can be affected by changes in temperature in the room, barometric pressure, large vibration sources. If you're in a city and the subway goes by or some other instrumentation is in the room and causes a lot of vibration, that can cause a problem. So the system will maintain constant sheath temperature to maintain a stable stream. But if there are any perturbances in the stream, the system will actually work to maintain and control that break-off point. So you'll actually be able to see this live stream image within your software, and you'll be able to see in real time the software making changes to what we call the drop drive and drop clock to keep that break-off where it was during that calibration. If it does get to a point where it thinks there's a bigger problem and it's worried that it can't maintain it, it will then give you a pop-up message that says the droplet condition has changed and it recommends that you redo the sort calibration for stable sorting. So this is basically a warning that, hey, things are changing more than I can maintain it. Go ahead and recalibrate. Or it's just going to tell you I cannot make the appropriate droplets for sorting, and then you can basically then stop your sort and do what you need to do to address that issue, whether it's purging air out of the system, or maybe you have a clog or something like that. But these fail-safe measures are there to ensure that you can sort for a long period of time and that you're able to troubleshoot the system in case of any instabilities. There are progressive warnings, like I said, before the stream is automatically stopped. But if there is a large issue, the stream will automatically stop. So in the event of a clog or an empty tube, the system will stop itself for those reasons as well. So that's super, but as the user, how do I know that I'm doing my part right? How do I know that I'm setting up my experiment properly? So when we come in and install the instrument and train you for the first time, we're going to provide you with all of these setup guides that will basically guide you from starting up the instrument, shutting down the instrument, through fluorochrome selection, through chip selection, through sorting mode selection. We have all these little quick reference guides and training outlines that we will provide for you, and that will help you set up your experiment properly. So we know the instrument is maintaining itself, but now we know that we're setting it up properly and we're giving ourselves the best, chances for success. All right, so we will shortly be launching version 3.3 of the MA900 software. And what's important about this is really supporting quick setup of different types of chips over the course of the same day. So we have basically tested, 30 different runs across 5 different instruments for each of these chip sizes. And we have found that each time the average calibration time is under 20 minutes. You can see here for the 100 micron, it's usually about 16 and a half minutes. The 70 is about 13 minutes. The 130 about 15 minutes. So we tried to test this to basically not only establish an average, but factor in, unit to unit variability. And this just illustrates the fact that even in a multi-application environment where you have people that want to run different cell types on different size chips, you can swap those chips out and have very minimal downtime before moving on to the next application. And again, this whole process is guided by a software wizard as well. So there's a click of a button in the software that says chip exchange. And then again, it will have you sort the new QR code and walk you through the calibration of the new chip. Okay. Here we basically did a test where we said, okay, let's see if this is really consistent over the period of a single day of sorting, right? And sort of switching back and forth between different chip sizes and different applications and ensuring that, in fact, yes, it is still quick over the course of a day and you still get good sorting, right? So we exchanged a few different sorting chips on the MA900 to ensure that switch and that recalibration is still efficient and that the system is set up in less than 20 minutes for any type of chip and that you still don't have that much downtime. So in this case, we start with a 100 micron chip. We sort some HeLa cells for viability and yield. Then we switch to a 70. Again, this takes about 12 minutes to set up. Then we sort some lymphocytes, again, for purity and yield. You can see here that all the data display still looks good. These are some post-sorting results where you can see that the purity and yield are still very good. And then we just switch back and forth each time under 20 minutes for the recalibration of the new chip. And then you can resume on sorting with that new chip and still get very, very good results. One other really, really nice feature is that if you have a user that comes in and they want to run a different cell type or a different application, but they want to use the same chip size, you don't actually need to swap the chip. So, you can have the previous user just clean the instrument, again, guided by a software little wizard where it will have you, run bleach and run water. And then you can actually log out of the system, check this little option here that says keep auto calibration result for next user. And then the next user can come in, log into the system, and not have to go through auto calibration at all because they're using the same size chip. All right. So in this case, you actually have zero downtime. The only thing you have, you want to make sure you clean the instrument for the next user. But again, in that multi-user environment where you're trying to cram in as many applications and make sure everyone has time to run their science as you can, this is a very, very nice feature that helps facilitate that. So in this case, is almost zero downtime. So with all these different applications being run, how can I trust that the instrument has been maintained and is cleaned and ready for my cells, right? Because I know that, I have a bunch of different people running all kinds of things through this instrument. And so I want to make sure that when I run my application, I can trust that everything's going to go to plan and I'm not going to have any contamination or things like that. So system maintenance and cleaning is also automated and guided by software wizards. The entire fluidic system is fully integrated with software-driven controls to simplify, setting up for an aseptic sort, cleaning the system, flushing the system with ethanol. These are all done by wizards and guided workflows. This fluidics cart that you see here that has your waste and your sheath fluids, as well as your cleaning fluids and your DI water. There's sensors in each of these little slots for each of these tanks. So in the software and on the front of the instrument, you'll see real-time feedback on your fluid statuses. And you're able to, basically manage all of these fluids right through the software. So it doesn't matter if you're sorting bacteria, mammalian yeast cells. You have the confidence that this system has been maintained and has been cleaned properly, because it is so easy to do. In addition to that, we talked about this earlier, but you can swap out that chip, and we'll talk about the sample line in a second, but you can swap out the sample line as well. And so you can actually clean the system and then swap out the chip, swap out the sample line, and now you have an entirely new sample path for your application, and you don't have to worry about carryover at all. Okay. So here's an example of what one of those guided workflows for cleaning the system looks like. In this case, we're looking at the ethanol cleaning wizard. But you can see here that these wizards are pretty intelligent themselves. So they have, little checkpoints in them. So in this case, the system is telling you that the bleach tank is okay and the ethanol and sheath tanks are okay, but the DI water and the waste tank have been removed. So in a lot of these guided workflows, the wizard actually will not let you proceed until you've done the correct thing, right? You've either filled or emptied the tanks, or done what you've needed to do, or what it's telling you to do. And then once you've done that, you can then click next. So in a multi-user environment, this is fantastic because oftentimes you get concerned about a user that doesn't have a lot of experience coming in and trying to do something on that machine and doing something incorrectly, and now you have to take time out of your day to remedy that situation. But in this case, this helps prevent that from happening in the first place. So not only is it easy to do, and you don't really need to write your own SOPs because it's all in here, but it prevents the wrong thing from happening in the first place. Again, so in that multi-user environment, this is super, super helpful for not only making it easy, but preventing those issues. And then this is just an example of the other maintenance and cleaning protocols available in the software. So we just saw the ethanol cleaning, but there's also one for bleach cleaning and priming, sample line exchange, like I mentioned briefly, as well as cleaning the sample area itself. So, opening up the instrument, taking the sample chamber apart, cleaning it all out. These are all guided workflows with pictures and instructions and checkpoints to make sure that things are done properly. And the user can just log in, pick one of these, hit start, and the wizard will begin, and they can perform that workflow on their own. In addition to having guided workflows, you can also customize them. So if you log in as the administrator, you have the ability to customize what is done during these protocols. For instance, how long is the bleach soak? Do you want to have an additional bleach cleaning or DI water rinse? And then you can also set reminders for these. So oftentimes, if you have a busy core lab or you have a multi-user environment where there's not one person responsible for overseeing the instrument, having some kind of reminder there to ensure that those are being done at a regular interval so that the instrument is being well maintained and cleaned is very helpful. So you can set these cleaning reminders to daily, weekly, monthly, and actually make them mandatory as well, which some labs like because then the cleaning must be done. All right, but this just adds that robustness to those maintenance and cleaning protocols to make sure that this instrument is always going to be ready for use. All right, so all of this is wonderful, but what happens when I run into a problem? And if you've ever run a cell sorter, you've probably run into a clog, right? So clogging of a nozzle on a traditional cell sorter, let's talk about what that looks like. So cells typically enter the flow cell and go through the nozzle without clogging. That's sort of the ideal situation, right? So here's your flow cell. You have your nozzle. Your droplets will flow down through these deflection plates. You have your waste catcher in the center, and then all of your individual populations are being caught in your collection tubes, right? So you have this high purity sort without much cell loss, and you also have minimal aerosol generation because everything's operating as it should, and things are going where they should. A non-issue. However, on most traditional cell sorters, the narrowest part of that path is the nozzle. And so that is where clogs typically occur, is in that nozzle. So when this happens, you now have a stream of droplets that is not going where it should. It's either missing the waste catcher entirely, it's pooling up on the tube holder, or you have non-targets going into your tubes. It's ruining your purity. Now you have, sheath fluid and other cells and whatever your sample is all over your deflection plates, dripping off your waste catcher. It becomes a mess. Aerosol generation is significantly increased. Now you have a potential biohazardous spill situation. And so this is just a pain in the neck, quite frankly, and it's something that everyone has dealt with. So on the MA900, this can be resolved by simply backflushing or priming the microfluidics chip. So that looks something like this. So as the sheath fluid goes in the chip, the sample fluid is injected here. The chip is designed in such a way that if a clog occurs, it typically occurs here in the chip. And so you're able to simply backflush the chip, get that clog out of there, and then resume flowing normally. And this all happens at the click of a button in the software. You do not need to take the chip out of the instrument. And again, you've minimized your downtime and you've allowed the user to resolve their own issue so that you don't need to get involved and things can resume running smoothly. Okay. So what if you have a clog in the sample line? Because the clog does not always occur in the chip. Sometimes it occurs in the sample line. Again, there's a software wizard, there's a cleaning wizard that will assist you in running a bleach and a DI water rinse at a high pressure to basically push that clog out of there, clean the sample line, and then again, you can just simply resume flowing your sample and resume sorting. Now, if you've got a really, really stubborn clog or some other issue with your sample line, the sample line can simply be exchanged again with a software wizard, right? So you go to that maintenance tab, you click on sample line exchange, and it will walk you through with pictures and instructions how to swap out that sample line. These come pre-assembled all in one piece. It takes maybe 2 minutes to swap it in and out of the instrument, and then you're ready to recalibrate and continue on sorting. So again, very minimal downtime, and you do not need to get involved with the issue, right? If you're the administrator or the person, overseeing these instruments, the user can simply go, oh, I've got a clog, and then attempt to clean it or exchange the sample line and probe for a new one. So the other benefit of having this exchangeable sample line is, like I mentioned briefly before, if you are extremely worried about sample carryover, you can simply either swap out for a new one or just bring your own sample line, put it into the instrument, and now you have a whole new clean sample path. You put a brand new chip in and you have no carryover concerns whatsoever. So like I said, in the event of a stubborn clog, or if you want an entirely new clean sample path for the next user or for yourself, very easy to swap this in and out. And what this means is that, like I said, it opens up your time as the manager or as the staff. It's because you don't have to deal with these issues. The user can deal with them themselves. And again, you're minimizing downtime on the instrument. So in a nutshell, what does all this automation mean? It basically means that any user can walk up to the system, set up for a sort, run a sort with confidence because it's being actively monitored by the instrument, and you know that when the next user comes down, or if you're the next user that comes down, that instrument is going to be clean, ready to use, and well maintained because all of these processes and steps and sorting are automated on the MA900. Okay. So there are many, many applications that have utilized the MA900 cell sorter, but let's focus on a couple of common ones. Okay, so we've seen neuroscience, we've seen stem cell biology, immunology, cancer research, but let's talk about some single cell isolation of cell lines, and we'll also talk about some immunophenotyping. So sorting large cancer cell lines for single cell isolation, something that we see a lot of labs do. It works extremely well on the MA900. This is an example of an osteosarcoma cell line that was sorted with the 100 micron chip. The software supports very precise calibration of the plate coordinates for plate sorting. So you can actually control the exact placement of the droplets, but you also have the ability to customize the size of the adjustments themselves, and you have the ability to lay out the deposition in any way that you'd like, even utilizing different populations and different sort modes within the same plate. PCR plate holders exist for 96 and 384 well plates that help ensure that the single cells are accurately deposited per well at a high efficiency. One of the things that helps with that as well is that our plate holders are angled. So that angle matches the angle of the side stream where your droplets are being deposited. And so this basically helps ensure that when you align the plate and you've got your droplet right in the center of the well, because that angle matches the angle that the droplets are coming out of the instrument at, you have a fairly high confidence that that droplet is going to go right into the center of that well, right in the bottom, and you don't have to worry about that droplet hitting the side and then evaporating and dying. We also have the index analysis feature. So this is something that is very helpful, where you are actually able to, once the sort has been performed and you've recorded the exact location of every single cell that was sorted, you can then go back and see this nice little plate layout of all the cells that you sorted. And in this case, say you're interested in wells A1 through B3, you can highlight those wells and see exactly in the data where those cells came from. And you can actually do the inverse as well. So let's say you're interested in a particular region of the population that you sorted. Maybe it's the brightest cells or some particular expression level. You can then create a new gate on those and select that gate and have it show up inside of your plate map. So you can see exactly where in the plate those cells of interest have landed. Okay. The second application we want to talk about is immunophenotyping. So this is an example of a 12-color TBNK panel where we've not only identified these rarer T gamma delta cells, but we've also been able to sort out the T cells, the B cells, and the NK cells with a very high post-sort purity, efficiency, and yield. And this is at a pretty high speed using the 70 micron chip. So this just illustrates that, on the same instrument, you can do a 100 micron chip, do a single cell sort from a cell line, and then swap out those chips with minimal downtime, go right into an immunophenotyping panel, do that very quickly, and still get very good results in both of those applications. Okay. So to summarize the MA900 platform, it is right at home in a multi-user, multi-application lab. Again, it's got a small footprint, fits right on the benchtop or inside of a BSC if you have biohazardous concerns. Easy to use. The automation, sensors, and cameras inside of this enable self-operation with many, if not all, of the workflows being guided with software wizards. It's extremely capable, 12 color capabilities supported by 2, 3, 4 laser configurations. You can sort into, up to 4 ways into tubes or into multi-well plates. And it truly is a 24/7 solution. So you can sort cells on your schedule, day or night, because any user of any skill level can walk up to this instrument, turn it on, scan a chip, set it up, and be ready to sort in a fairly short amount of time. Okay, so if you have further interest in the MA900 and you want to learn more, you can head over to our website. We have a virtual product tour that you can access via the website. We also have other resources like brochures, publications, webinars. That's all available at sonybiotechnology.com. I want to thank you very, very much for your time and attention, and I'm happy to take any questions.
Thank you for that very informative presentation, Anthony. It's now time for our question and answer session. So if you have any further questions, please do send those in. You can submit these by typing into the questions icon to the left of your screen. And the first question we have for today is, what is the largest and smallest size cell that I can sort with the MA900? Can you sort tiny cells on a big nozzle? And is there a higher chance of getting two small cells in one big droplet if I use a very large nozzle?
That's a great question. So you generally want to use a nozzle size that's about 3 to 5 times larger than your cell size. But more specifically, the largest cell type is about right around 30 microns. That we have going up to 50 microns with gel beads specifically. And the smallest is about half a micron, 0.5 microns. To answer the second question, you can always sort smaller cells on a larger nozzle. That's not a concern. Generally, you're not going to have any clogging issues or anything like that. But typically with smaller sample sizes, your concentrations tend to be higher. So yes, there is a chance you could end up with more than one cell within those larger droplets. But depending on your sort mode, those droplets will typically be aborted and sent to the waste.
Great, thank you for that answer.
Yep.
So moving on to our next question here, we have, how do we clean the chip between sample types?
Also a great question. Very easy. What we recommend people use is just 10% bleach and then DI water or MilliQ water, whatever the cleanest water you have available to you. And again, this can be done with a wizard, or you can just run them, as samples on the instrument before you swap out the chip.
Excellent, thank you. So moving on to our next question, we have, can bleach go through the nozzle since it's all in one piece?
Yes, absolutely. Yep. Just as I mentioned earlier, you can run bleach through the nozzle. It's sort of our recommended cleaning method, and it's also what we're going to do to recommend to you for your daily shutdown. So when you go to shut the instrument down at the end of the day, we're going to say run bleach and run water through that sample line and through the nozzle.
Perfect, thank you very much for that answer.
Mhm.
So our next question is, what kind of instrument maintenance is required to keep the MA900 running smoothly?
Good question. Honestly, the instrument maintenance is fairly simple. Like I just mentioned, there's pretty much the daily cleaning, which is going to consist of bleach followed by water, and then the instrument will shut itself down. And then more long term, there is a monthly ethanol cleaning that we recommend you do. Again, this is all wizard driven. It's going to have pictures and instructions to sort of walk you through that process. But this monthly ethanol cleaning will have ethanol flush through the system, cleaning the entire fluidics path. It will sit overnight in ethanol, and then you will flush out the ethanol the next morning, and then reprime the system with PBS.
Great, thank you. And it's great to see lots of great questions coming in. Reminder to our audience that if you do have any further questions, please do send those in. You can submit these by typing them into the questions icon to the left of your screen. So now moving on to our next question. This is, does higher sample pressure cause loss of cell viability? Is there an ideal sample pressure which I can use for 70 micrometer nozzle?
This is a great question. No, higher sample pressure does not really cause loss of cell viability. That's kind of a misconception. Really, the sheath pressure is what's going to be a larger concern. That's going to be dictated by your nozzle size. But the sample pressure is not going to have that big of an effect on your cells. In terms of an ideal sample pressure, this is going to be sort of dictated by what the optimal event rate is for that nozzle. So in the case of the 70 micron nozzle, you want to try to keep your event rate between 10 to 15,000 cells per second. So you can sort of adjust your sample pressure freely as the sample is running to keep the event rate, in that range. But you also want to consider maximizing your efficiency. So I don't really want to give a specific, like sample pressure number or anything like that. It's all about the events per second for that nozzle, and then your efficiencies.
Excellent, great. Well, it looks like that's all we have for today. So I'd like to thank our expert speaker again for today's informative presentation and discussion. And a big thank you to everyone joining us online. We hope you found this a worthwhile session. If you do have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speaker. And not to worry if we didn't get around to answering your question today, we will follow up with these via email after the webinar. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us today.